Recombinant proteins in medicine. Hello, my name is Rory, and with the help of Sandra and Eugene, we'll be discussing the use of recombinant proteins in medicine. I will first discuss what recombinant proteins are used for in the medical field, the history and development of the technique, and how the process works. What are recombinant proteins? To understand recombinant proteins, we first have to look at recombinant DNA as a whole. In extremely simple terms, recombinant DNA or rDNA molecules are DNA molecules that are formed via laboratory methods of genetic recombination, such as molecular cloning, to combine genetic material from multiple sources. This allows for the creation of sequences that would not otherwise be found in the genome. Recombinant proteins, then, are created from cloned DNA sequences which encode for a specific protein with a known function. How are they used in medicine? This technology is extremely useful in the field of medicine as it allows for not only the treatments of diseases by replacing damaged and diseased genes with new ones, but also allows for the production of specific hormones and vaccines. The majority of human diseases are systematically or partially related to the dysfunction of specific proteins, meaning the demand for recombinant proteins for therapeutic applications is ever growing. Therapeutic proteins provide important therapies for diseases such as cancer, haemophilia, diabetes, anemia, and infectious diseases. However, I will leave these to be discussed by Sandra in the current applications section of our presentation. History and development of the technique. The first recombinant molecule that contained DNA from different organisms was developed by Paul Berg in late 1971 in his laboratory at Stanford University. This landmark was quickly followed by another, when in 1972 Herbert Boyer and Stanley Cohen successfully inserted rDNA into bacteria in such a way that foreign DNA would be replicated naturally. In 1972, researchers such as Boyer also began to realize that the enzyme EcoRI cut DNA in a way that the ends were staggered, as opposed to blunt. This meant that no molecular additions were needed to make one piece latch onto another piece with complementary cuts. Boyer, with the help of colleague Robert Helling, attempted to insert the bacteria E. coli by using EcoRI to open up the bacterial virus Lambda. They were, however, unsuccessful as the enzyme continually cut the DNA in 5 places as opposed to the desired one. 1973 saw the first successes of the famous Boyer-Cohen collaboration, involving the use of one of Cohen's plasmids, PSC-101. This plasmid allowed E. coli to be resistant to the antibiotic tetracycline. The plasmid was cut at one site by EcoRI, allowing the plasmid to still replicate. This plasmid was then combined with other DNA that had been cut by the same enzyme, with the complementary ends of fragments from both sources joining to create new loops. The DNA was introduced into a calcium chloride-treated bacteria and then spread onto a culture containing tetracycline. Only the rDNA plasmid survived. It was a success. The pair progressed onto proving that genetic material could in fact be transferred between species. They snipped a piece of plasmid responsible for staph infection and spliced it with an E. coli plasmid. The DNA from the staph infection bacteria successfully replicated in the E. coli, which is a different species of bacteria. This progressed to the insertion of genes taken from the South African clawed frog into E. coli, an even more impressive success. These findings began to spur the beginning of commercial startups, with it soon becoming apparent to scientists that it was feasible to use bacteria in which human genetic information was incorporated to duplicate the body's natural ability to fight diseases, as well as remedies to birth disorders. The first major success of this was when Boyer and his colleague Kiichi Itakaru successfully expressed the hormone somatostatin in bacteria in 1977. This hormone plays a major role in regulating growth hormone, with recombinant somatostatin being shown to be virtually identical to its naturally occurring counterpart. 1978 saw continued success for the pairing, successfully constructing a plasmid that coded for human insulin. The foundations for recombinant DNA technology were now in place. The industry continues to grow, but we now see over 170 recombinant proteins being produced and used in medicine worldwide. How does recombinant DNA work? The key steps of creating recombinant DNA, and in turn recombinant proteins, are isolating of the DNA, cutting the DNA, and amplifying the recombinant DNA. Isolation. The first step in making recombinant DNA is to isolate the DNA donor and vector. DNA is extracted from the donor organism and will be the nuclear genomic DNA in eukaryotes or the main genomic DNA in prokaryotes. A bacterial plasmid is then harvested to be used as the vector. Cutting the DNA. The key moment that made recombinant DNA technology possible was the discovery of restriction enzymes. These are the enzymes that act as scissors, cutting at specific DNA target sequences, making them perfect for DNA manipulation. Let's look at an example we discussed before using the restriction enzyme EcoRI from E. coli. EcoRI recognizes this 6 nucleotide pair sequence in the DNA of any organism. The enzyme EcoRI cuts a pair of staggered cuts between the G and A nucleotides. This cut leaves a pair of identical single-stranded sticky ends, meaning that they can bond to a complementary sequence. The basic principle is that if two different DNA molecules, such as the vector DNA and the donor DNA, are cut with the same restriction enzyme, both will have fragments with the same complementary sticky ends, making it possible for the fragments to combine. While the sticky ends have now joined to generate a population of chimeric molecules, the sugar phosphate backbones of the molecules are not sealed. This is amended by the addition of DNA ligase, which creates phosphodiester bonds at the junctions, sealing them, shown in the image below. Amplifying recombinant DNA. The ligated recombinant DNA now enters a bacterial cell via transformation, where the vector is now able to replicate due to plasmids having a replication origin, effectively a tendency to replicate. The key part of this is now that the donor DNA insert is a part of the vector's length, it is too now automatically replicated. Many cycles of cell division will take place, resulting in a colony of bacteria that will contain billions of copies of the single donor DNA insert.
Recombinant DNA technology has been widely used in medicine for diagnosis of genetic disorders and identification of infectious diseases. It allows isolation, multiplication, or expression of the insert target gene without having to synthetically create biological molecules. Therefore, it is not only effective for treating many diseases such as diabetes, but it is also inexpensive. For instance, human growth hormone is a protein made from a chain of amino acid subunits. Prior to development of rDNA technology, human growth hormone was only able to be produced by isolating it from pituitary gland tissue that was taken from human cadavers. This process was time-consuming, expensive, and sometimes unsafe due to contaminants from cadaver tissues. This may result in human growth products occasionally containing contaminants, and rarely patients injected with human growth hormone from cadavers developed Creutzfeldt-Jakob disease, which is a very serious human version of mad cow disease. rDNA technology has eliminated these potential contamination problems. Additionally, various rDNA techniques exist which offer different advantages to traditional plasmid-based systems. For example, chromosomal integration of the foreign gene allows for animal cells to be used as the host, which results in higher stability. This increased stability is a result of animal cells being more similar to human cells than bacteria, because animal cells are also eukaryotic rather than prokaryotic, like bacteria. Also, viral vectors are an alternative rDNA technique which uses viruses to infect the host with foreign DNA. Viral vectors make the process of inserting DNA easier and more rapid than plasmid-based. Therefore, it is more viable for industrial production of recombinant proteins. The widespread industrial usage of the insect cell baculovirus expression vector system, also known as BEVS, is a good example of the commercial success of the technique. BEVS is also suitable for the production of many vaccines.